this video, we'll cover bacterial flagella structure. We will start with the gram-positive bacteria because they are simpler. You can see the flagella have several components that anchor them to the bacterial cell membrane and let it pass through the cell wall. In the cell's membrane, a structure called the S-ring holds a rod in place. This rod acts like the axle of the flagella, and it is spun by the motor protein called M. The rod extends through the cell's periplasm and through the cell wall using a third ring called the P-ring. Outside of the cell wall, the rod is connected to a hook which is attached to the filament of the flagella. This acts as the propeller itself. The next diagram shows a gram-negative bacteria, and it has one additional ring, called the L-ring, to accommodate their outer layer. That is because there is LPS in this layer. The filament at the end of the flagella is composed of many flagellin proteins, which are arranged into a helix. When the flagellum grows, flagellin molecules are moved through the hollow rod into the filament. The flagellum cannot grow from the base of the filament because that would cause it to detach. Instead, the flagellin proteins move all the way to the tip of the filament and are attached there. Here is another demonstration of how the bacterial flagellas grow. This is a reminder that they do not contain microtubules like the eukaryotic flagella. Instead, they are composed of the protein flagellin, which you will see repeatedly written on the streamer. This is to represent all the flagellin proteins coming together to form a long helical structure. When a new piece is added, it travels through the hollow center of the helix, which is represented by the toilet paper roll, to the tip of the filament structure. Here we see another overview of the bacterial flagella structure. We will review some parts, but also talk about more parts in detail. This specific image shows a gram-negative bacteria because it has the L-ring in the LPS layer. The P-ring is in the peptidoglycan layer. You can see the S and M rings located in the cell membrane. The S-ring, or the switch, is what determines the direction the flagella will spin, such as clockwise or counterclockwise. The M-ring, or the motor protein, is what spins the flagella. We already know the flagellum is a helix, and because of this, if the rotation is clockwise, the bacteria will begin to tumble. The helix coils tighter and becomes tangled, causing the bacteria to uncontrollably move in a random fashion. There is no clear direction, and it can't control where it is pointing when it comes out of a tumble. When the flagella begins to spin counterclockwise, however, the flagella then streams out behind the bacteria, causing it to run, or swim. The energy to do this comes from the proton motive force, not ATP. Naturally, bacteria will swim for 1 second and then tumble for 1/10 of a second. However, if there is a chemoattractant around, the bacteria will swim for a slightly longer time, making the overall net movement closer to that. In these examples with the ceiling fan, note the bacteria can have more than one flagella, and each one will have its own basal body ring attachment system. But in this case, only one basal body was shown. The other flagellas were just for demonstration purpose only. I hope you enjoyed this video and found it educational. Here are our sources. Thanks for watching!